My name is Mitsuko from Harvard University. So information for both reward and threat is important for animal survival. How does the brain appropriately predict the potential reward and threat? And how does it balance those information? So theoretically, many reinforcement learning models do not distinguish the types of outcomes. So so that those models map those model maps everything, including reward and penalty, onto the one-dimensional value axis. And then the dopamine neurons send reward prediction error, comparing the outcome value and the prediction, and other brain areas learn to predict outcome value and facilitate action to maximize future rewards and minimize future penalties. However, more recent studies suggest a more complicated view, because some dopamine neurons are excited with the non-rewarding stimuli, such as aversive stimuli and self-movement. So in my talk, I will focus on reward and threat and discuss how we can reconcile the classical reinforcement learning model by incorporating diverse dopamine systems. So we recently found that dopamine neurons that project to the tail of the striatum receive unique set of presynaptic information. So these are the presynaptic distribution presynaptic neurons. And from this screening, we found that these dopamine neurons are anatomical outliers among the 8 subpopulations of dopamine neurons we tested. So we named this area tail of the striatum, or TS, because of the anatomical similarity to the caudate tail in monkeys. So these screening steps suggested a unique function of TS projecting dopamine neurons. So TS is also called the sensory striatum because they receive visual and auditory cortices and thalamus, and also the temporal cortices. In humans, TS is right above the hippocampus and right posterior to the amygdala. And because of this location, people believe the previous fMRI imaging studies probably categorized TS as hippocampus or amygdala. So we recorded these dopamine neurons in mice. So in normal, the dopamine neurons respond to water, and the response is modulated by water amount, consistent with the idea of reward prediction error. So these dopamine neurons, the TS projecting dopamine neurons, do not really encode valence and respond to various external stimuli in the world. And the responses are modulated by the intensity of these stimuli. So this suggests that there's at least two axes of the evaluation systems in the dopamine system. One along the value axis to signal aversive and rewarding stimuli, and one along the intensity of the external stimuli. So to know the function of these dopamine neurons, before that, sorry. So also the prominent feature of these dopamine neurons is that, so these dopamine neurons respond to the novel stimuli very, very strongly. And then the response decay with repetition of the stimulus presentation. So to know the function of these dopamine neurons, we tested the novelty exploration. So in this task, so the novel toy is presented to a mouse in a familiar environment. And normal mice show this kind of the approach and quick retreat many, many times. In contrast, so after ablation of TS projecting dopamine neurons, mice started to lose the timidity toward the novel toy much quicker than control animals and show object engagement. So to know this behavior, to examine this behavior in more details, so we use the machine learning technique, the recent machine learning technique like MOSIC, to segment the behavioral structure, and then also deep labka to track the body parts. Using this, combining both of these techniques, we found that two behavioral motifs or syllables are enriched when animals encounter the novel object. One syllable, 79, was represented the cautious approach, and another syllable, 14, represented rapid retreat. So this syllable 79 was frequently followed by syllable 14. And surprisingly, after ablation of TS projecting dopamine neurons, the transition frequency didn't change. So we separated approach types into two types, the first investigatory approach and then engagement. So we found that all the animals we tested showed investigatory approach at the beginning, but the length of the investigatory approach was the variable across animals. And then after investigatory approach, some animals showed the object engagement, and some animals showed the total avoidance. So what determined whether animals engage or avoid after investigatory approach? Ablation of TS dopamine neurons slightly decreased the investigatory approach, and dramatically increased engagement. So because this is an effect of ablation, so endogenous TS dopamine is suppressing engagement, the potentially signaling threat prediction. So the animals might learn the threat prediction during investigatory approach to decide whether they engage or avoid. In this way, so this initial approach behavior might be interpreted as risk assessment. So indeed, when we look at the behavior and TS dopamine activity during risk assessment, we saw that every time when animal came to the closest point with the object, we saw the higher activity of these dopamine neurons. Something like looking like that they are sending the evaluation signal. So in this way, these behaviors are actually natural behavior without any rigid task structure, but animals naturally form the trial-like structure to allow the incremental learning. Okay, so from this, their trial-like structure suggested so that the naturalistic, very complicated behavior might be modeled by a simple reinforcement learning model like this. However, to use this kind of learning, animals first know, or the brain has to know what is the threat. So before going to the definition of threat, I'm going to tell you about the various ideas about the word. So the inferential theory by Hall proposed that all the motivation comes from reproduction success, and thus the physiological needs, such as the eating good food or the avoiding harm. However, it is often difficult and time-consuming to really confirm whether the core physiological needs are fulfilled or not. To overcome this problem, the brain might be used as subcores. In psychology, if you train animals with a complicated task, Instead of starting from difficult tasks, experimenters usually start from the easier task and then gradually make them to do the more complicated task. So the reward is given at the gradually, after completion of gradually longer sequence of actions. So this method called shaping in psychology is very powerful, and because it overcomes the problem of sparse reward when the achievement of the final goal is difficult and very commonly used to train animals. So now, in the brain, so there might be the two strategies to use subgoals. The first strategy is similar to Skinner's shaping. You might assign reward to subgoal. However, the reinforcement learning works to predict the summation of all future rewards. So adding additional rewards as subgoals always skew the learning. So potentially the animal may not learn the optimal action to reach to the goal. So in the extreme case, the animals might just learn to get subgoals because it's reward. Another strategy to use subgoal is the optimistic initial value. So optimistic initial value means that animals initially optimistically predict reward at the subgoal. So it's not reward itself, it's a reward prediction. So in this case, because reward prediction can be updated by learning, so the reward prediction at the start can be converged to the actual outcome value. And then still, this method can promote the exploration and learning. So this method is powerful and then often used in machine learning technique. So using this idea of the optimistic initial value, and then this is also called the shaping bonus because it shapes learning by promoting exploration. And using shaping bonus, so recent studies suggested that the eventual goal of eating is to become the big, strong, and healthy, but the brain might be used the nutrition or even flavor to induce the approach and learning. And the recent study also showed that you will hear the more detail in the afternoon, so don't worry. And the recent study even showed that the drinking also can be evaluated at multiple steps, like the flavor and blood osmolarity. So in this sense, recent multiple studies are arguing whether the dopamine responses at the reward onset should be increased or decreased. But at the brain's point of view, the reward onset at the task or by the experimenter is just the start of the cascades of the reward prediction at many physiological level. So whether the reward prediction at the reward onset will increase or decrease depends on the various factors, such as the animal's history of the training or the task, and also the individuals. So what about threat? So how the brain defines threat? So as I mentioned, the dopamine neurons project to tell the striatum show the response to the novel stimuli and the response decay if the cue is not associated with anything, or if the cue is associated with reward. You can see the decrease. However, if the cue is associated with the threatening stimuli, such as air puff to the eye, the response stays high, and then here. So this suggests that the threat prediction is initialized high and gradually decay if animals confirm safety. So this suggests that some similarity with the idea with the food reward So the ultimate goal of the threat prediction is to predict harm. However, an individual cannot really learn from this, and it's very catastrophic if they only learn from injury. And so brain might have some strategy to predict the potential threat using physical science. So physical science is very easy, and the brain might use physical science to predict the potential harm. So we model threat prediction rather than the reward prediction. So we modeled novelty exploration using the shaping bonus, that is the threat prediction at the object. So this means that when animals approach and then encounter the novel object, they think that something bad might happen. We used temporal difference learning, or TD learning, it's called TD learning, and threat prediction moves forward to earlier and earlier time point. Using this model, the threat prediction at decision point will increase at the beginning because of the initial threat prediction at the object, but decrease and eventually to 0 because actually the object is harmless. We also considered uncertainty to have the more practical range of threat prediction. So you cannot see very well, but there is some range to show the threat prediction using threat out uncertainty. So to make decision to choose their action, animal might just compare their threat prediction and threat threshold. So if threat prediction is higher than threat threshold, animals avoid. And if the threat prediction is lower than threat threshold, they might engage. If the threat prediction is uncertain, they might do the risk assessment. So I will show you what kind of behaviors this model will predict. So basically, this model predicts that the individual variability and also the complicated temporal dynamics along the time. So the shaping bonus can be variable if the object is different. So if the object is salient, the shaping bonus is high. That's the definition. However, even for the same object, it depends on the animal. So if sensitive animal might perceive the object salient than others. So if the shaping bonus is small, animal may first show the risk assessment because of high uncertainty, but quickly move to the engagement. If the shaping bonus is intermediate, they may show the sustained risk assessment and then engage. If the shaping bonus is high, animal may first show the risk assessment, but then avoid. After avoiding, they lose the learning anymore, and then trapped in avoidance state, so showing the sustained neophobia. So in this way, our model predicts that neophobia may come from the general sensory hypersensitivity, such as in the autistic patients with sensory hypersensitivity. So our model have several insights, and then first, we modeled threat prediction instead of reward prediction. And to do that, we incorporated the idea of shaping bonus and uncertainty, which is commonly used to model reward prediction. And another point we want to point here is that we modeled using the very, very simple structure of the reinforcement learning, we can explain the diverse and diverse behaviors across times and also across the individual variability. Okay. So these studies suggested a separate evaluation system of the threat prediction, apart from the reward prediction. But the algorithmic structure is very similar. So now, separate systems might cooperate, but also compete each other. So we tested this idea next. So in this paradigm, we call monster paradigm. Animals just get water and come back. That's it. However, sometimes there's a monster. So here is a monster, and mouse come out. And without drinking water. So similar to the head fix task, we found that the dopamine response to the monster depends on the size. So if the monster is big, the response to the dopamine is high. And then behaviorally also, the animals, of course, avoided big monster much more. And we did the causal manipulation. And then if we ablate TS dopamine, the avoidance of big monster is decreased. And oppositely, if we increase the dopamine in the naturalistic level, and then the animals avoid even the mini monster that's smaller than themselves. Okay. So our study showed that there are at least two separate dopamine evaluation systems. And then these systems might support has several advantages, but I will point only 3 points. So 1 point is, because this is independent, separate system, they can prioritize one system depending on animals' external and internal states. For example, when animals is starved, the brain might prioritize this system. And another advantage is they can have the shipping bonus, the threat prediction can have the physical science, and the reward prediction system can use the flavor to make the initial prediction. Maybe I can I can find another. What was that? Okay, I'll talk later in discussion. Okay. I will thank all the lab members, including now for continuous encouragement, and Iku, Koriki, and Will for the project in this talk. Thank you so much.